Today we are here to discuss about Mycobacterium tuberculosis, the causative agent of the deadly disease TB or tuberculosis. Now, TB has affected human beings for centuries. Historically, it was given different names such as consumption, wasting disease, white plague, etc. TB killed millions of people. So, government sponsored some program to get people concerned about TB. For example, here you can see a stamp published by the Red Cross in 1924. If you buy this stamp, then you are contributing to the TB control program. Till mid 1800s, most of the people believed that TB was hereditary. Okay, so it belongs within a family and transmits from generation to generation. But then in 1865, Jean-Antoine Villemot proved that TB was actually contagious, not hereditary. Then the major finding came in 1882 by Robert Koch, the famous German scientist, who discovered the causative agent of the TB disease, the bacteria Mycobacterium tuberculosis. So he was awarded Nobel Prize for this discovery in 1905. Here you can see a scanning electron micrograph of Mycobacterium tuberculosis. It's a rod-shaped bacteria. Before the discovery of the anti-tuberculosis drugs, most of the patients, they were sent to places called sanatoriums. Okay, so there the patients followed a particular regimen of bed rest, open air and sunshine as shown here, accompanied with nutritious food. But sanatoriums were quite expensive. So people who could not afford sanatoriums, they had to stay at home, and many of them died without any treatment. There are some ineffective treatment were available with some patented medicine and vaporizers of chrysoline, but they were useless. So the major breakthrough, that is the discovery of a drug that could kill Mycobacterium tuberculosis and thereby stop the infection and the TB disease, those drugs, they were discovered in the 1940s and 1950s. Here some drugs are shown: isoniazid, rifampin, pyrazinamide and ethambutol. Streptomycin was also there, but is not shown here. Now, streptomycin was the first anti-tuberculosis drug discovered. It was discovered in 1943. And then, in the following years, between 43 and 1952, other two drugs were discovered, isoniazid and para-aminosalicylic acid. And in the following years, many other drugs were discovered. So, these drugs were quite effective at that time. As a result, TB death rates began to drop very fast. And each year, we have fewer and fewer cases of TB infection. And the success was so good that the sanatoriums were almost out of business, and most of them were closed by the mid-1970s.
Sir, does that mean TB was completely eradicated by this time? And if so, then why we are still studying it?
That's a very good question. The fact is that by this time people thought that TB is finished. Just like smallpox was gone for a long time. Then suddenly tuberculosis shocked the human community by re-emerging about mid-1980s. It was so shocking that, and a lot of people are discussing about it. It came in the cover of many famous international magazines as well. There are many factors that contributed to this resurgence of tuberculosis. For example, since people believed that, well, TB is gone, so the funding for TB control programs were not that good. That's one thing. Secondly, multi-drug resistant TB, they emerged, they increased and they spread. That's another reason. And the third reason was that the increase in the number of HIV infected people. We'll discuss it later. So the governments all over the world, they were alarmed, and they started funding to these TB control programs. As a result, in the beginning of the mid 1990s, the occurrence of tuberculosis disease and mortality started to decline. And fortunately, in the global level, it is still going down. Here you can see prevalence is going down, incidence is also going down, mortality is going down. So overall, this is kind of promising observation that with the improved and well-funded TB control programs all over the world, the TB cases are going down. But there are two things we should remember. First of all, this is the overall global pattern. But individually, some countries, in particularly say India and few other countries, the situation of TB decline is not so promising. Okay, so we have to be more careful. And second of all, we have already seen that TB has surprised us in the mid 80s. So it might so happen that it may again re-emerge in future. So we have to be very careful, and we have to maintain these TB control programs carefully. So this kind of summarizes the TB history timeline. So first, it was proved that TB is contagious. Then the bacteria causing TB was discovered. Then sanatoriums were established. Then the first anti-TB drug, streptomycin, was discovered. Then other drugs were discovered. So this resulted in good decline in the TB cases. So in the mid 1970s, most of the TB sanatoriums were closed. Then again in mid 1980s, TB re-emerged, and followed by good TB control programs, which resulted in decline in the TB cases from 1993 onwards till date. Now, the transmission of tuberculosis, that is spread from one person to another, of course, is mediated by the causative agent Mycobacterium tuberculosis, which causes most of the TB cases. There are other mycobacteria that cause TB, or I should say TB-like disease, like Mycobacterium bovis, Mycobacterium africanum, Mycobacterium microti, Mycobacterium cannati, etc. Here, this is a acid-fast stain image of Mycobacterium tuberculosis. These are the bacilli. There are many other Mycobacterium species that do not cause TB. For example, this Mycobacterium avium complex, non-tubercle bacilli. This transmission is actually mediated through air via droplet nuclei. Okay, so when an infectious person expels Mycobacterium tuberculosis, then these droplet nuclei are released in the atmosphere, which contains the infective Mycobacterium tuberculosis. Okay, and these can happen due to several reasons, like coughs, sneezes, speaks, and even sings. Actually, singing releases most of droplet nuclei compared to other. Now, transmission occurs when another person inhales this droplet nuclei. So here it is shown in this simple animation. You can see this person who is infected with TB, and here you have the bacteria in his lungs, and when he is releasing it, the droplet nuclei are traveling, and this person is inhaling. Then the droplet nuclei containing the bacteria goes to the lung, and then again causes the disease. So the probability that TB will be transmitted depends on several factors. First of all, the infectiousness of the person with the TB disease. Second of all, environment in which the exposure occurred. Say if the environment is a closed one and humid one, then the chance of transmission is higher. Obviously, length of exposure. The more prolonged the exposure time, more the probability of getting transmitted. And definitely the virulence or the strength of infectiveness of the tubercle bacilli concerned. That also plays quite important role. Not all Mycobacterium tuberculosis strains are equally effective in this. Some are more, some are not so. The best way to stop transmission is, the first step is isolate the infected person and also the infectious person. So if you isolate that infectious person, that means the chance of transmission to other healthy people is minimized. And second of all, start that therapy of that person immediately. Okay, so if that person is cured, then obviously he or she won't be able to transmit the disease. Now, this problem with drug resistant tuberculosis, when it started, people worked on it, found that it is caused by Mycobacterium tuberculosis strains that are resistant to at least one of the TB treatment drugs. So these 4 drugs, they are the first line drugs: isoniazid, rifampin, pyrazinamide, ethambutol. If the bacteria is resistant to any one of them, we will call it a drug resistant Mycobacterium tuberculosis. And of course, resistant means that particular drug won't be able to kill the bacteria. Now, this drug resistance can be viewed from different perspectives. First of all, we can think of primary resistance, which is caused by direct person-to-person transmission of these drug-resistant bacteria. Then we can think about secondary resistance that develops during the TB treatment. Okay, there are 2 ways. First of all, if the patient was not given appropriate treatment regimen, maybe the dose was not proper or the combination of antibiotics was not proper. And another way is maybe the patient did not follow the treatment regimen as prescribed. Maybe that patient did not complete the dose, something like that. Then another way of viewing resistance is we define resistance according to the extent of resistance against the antibiotics. For example, it can be monoresistant if it is resistant to any one TB treatment drug. It can be polyresistant if it is resistant to at least any 2 TB drugs. But you have to remember, not both isoniazid and rifampin, either of these. Then there are multi-drug resistant tuberculosis, resistant to at least isoniazid and rifampin, the best first line TB treatment drugs. So this time resistant together. And finally, the extensively drug resistant tuberculosis. Okay, that's the most dangerous strain, resistant to isoniazid and rifampin, plus resistant to any fluoroquinolone antibiotic, and at least one of the 3 injectable second line drugs like amikacin, kanamycin, or capreomycin. Now, the pathogenesis, that is the development of the TB disease, that's also a very interesting aspect. Sometimes it may happen that the bacteria has entered the body of an individual, but no disease manifestation is there because the immune system is keeping the bacteria under control. Here there will be no symptoms, and we call it LTBI or latent TB infection. So it can be detected by the tuberculin skin test or the Mantoux test, or by blood tests where you detect interferon gamma release assays, or in short IGRAs. They are of several types like quantiferon TB gold test, quantiferon TB gold in tube, and the T-spot TB test. We will discuss the mechanisms later. It is important to know that people with LTBI are not infectious. The development of LTBI to the proper tuberculosis disease, that is TB disease, occurs when the immune system cannot keep the tubercle bacilli under control. Now, this development may happen very soon after infection, or it may happen many years after infection, depending on how long the LTBI remains latent. It has been seen on the average about 10% of people with normal immune systems who have LTBI will develop TB disease at some point in their lives. In contrast to LTBI, people with TB disease are mostly infectious. So this is the mechanism in detail. So the tubercle bacilli is inhaled through droplet nuclei and enters the lungs, where it travel to the alveoli of the lungs, that is the small air sacs. So this is a magnified view of the alveoli, where you can see the tubercle bacilli is here and it can multiply inside the alveoli. Okay, that is the beginning of the infection, we can say. Now from here it can travel via the bloodstream to other parts of the body. It's not always the case, but it may so happen. So from lung it can move to, say, brain or bone or kidney and few other areas. And then it can go to those new places and infect there, that is multiply there. But one common observation is wherever the infection is, as soon as the bacteria starts multiplying, there is an immune response such that a number of macrophages, a certain type of immune cells, are recruited to the site of multiplication. Here it is shown in the alveoli, but it is true for any other infected areas. So these macrophages, they form a shell-like barrier around the bacterial population. The result is that the supply of nutrient and everything is blocked to the bacteria. So the bacteria stops multiplying. It does not have any food or no oxygen supply. So the bacteria is kind of contained here in an inactive manner. So it is not doing anything, just means staying there. And this barrier actually holds or contains the infection in this place. So this is the situation of LTBI, latent tuberculosis infection, because since the bacteria cannot multiply and cannot spread to the other parts of the body in this situation, so it cannot manifest the real TB disease. It happens within 2 to 8 weeks of infection. Sometimes what happens is that this barrier is broken and then again the bacteria will receive oxygen and blood supply. They will start multiplying and then they can also travel to other parts of the body. So this results in rapid development of the TB disease. So this happens when the immune system fails to control or contain the bacteria in the site of infection. We can compare in this way the LTBI versus the real TB disease. So in LTBI, the bacteria is inactive, not multiplying, and in case of TB disease is active and multiplying. But both will show positive test if we do the tuberculin skin test or the blood tests as we have described before. Chest x-rays, they are usually normal in case of LTBI but show some abnormality in case of the TB disease. Sputum smears and sputum cultures, they show negative result in case of LTBI, but with the TB disease they show positive results. In LTBI there are no symptoms, whereas in TB disease you have symptoms like cough, fever, weight loss. And LTBI is not infectious, whereas the TB disease is mostly infectious before treatment. So when people get exposed to TB, the resulting situation can be two types. First of all, the person though exposed to TB will remain not infected. Obviously that person will also remain not infectious, and the result of the tuberculin skin test or the blood test will show negative result, and the conclusion is no TB infection. But some other persons can get infected, then the situation will be LTBI, latent TB infection. Again, the person will remain not infectious, but then in contrast to this situation, if we perform the tests, that person will show positive results. So we will conclude latent TB infection. Immediately the proper regimen of treatment should be started, because otherwise we have seen that the LTBI may go on to develop the actual TB disease.
Sir, does the nature of development of TB disease vary from person to person?
This is a very interesting question. We call these risk factors, okay? There are certain factors which increases the risk of development of TB disease from LTBI, okay? There are many factors. We will discuss two major factors here. One is diabetes, another is HIV infection. So when there is TB infection and there are no risk factors, then the risk is low. It's about 5% in the first 2 years after infection and about 10% over the lifetime of the individual. So it is not very high. But when there is TB infection along with diabetes, then the risk becomes 3 times higher. Okay. So overall, we have about 30% of risk of development of TB disease from LTBI. And finally, the highest risk factor, which is HIV infection. In this case, if TB infection happens, then the risk is about 7% to 10% per year. So in the overall lifetime, the risk is very high. As you can see, almost all the individuals, if they have TB as well as HIV, they will show TB disease. Okay. The reason is that HIV results in destruction of the immune system. So now the bacteria will be able to multiply at an abnormally high rate and show the TB symptoms very fast. So most of the HIV patients in India, for example, they die in TB. So this will be the end of our discussion today. If we summarize, first we talked about briefly the history of tuberculosis, and then we moved on to the transmission of TB, how it spreads from one person to another. Then we discussed about the various aspects of drug resistance, what are the different types, etc. And then we talked about the pathogenesis, the latent tuberculosis infection versus the actual tuberculosis disease. And finally, we talked about the mechanisms by which the LTBI can develop to the tuberculosis disease.